All right, going live, going live, going live, going live. I am live. I am live. All right, guess what? I am live. Guys, check the new, check the new link. The new link. I can't delete this. You know why I can't delete it? It has so much in it. So it is a blaze. Well, I have to write it on something and show them. No, so then just a chat.
Put it down. Put it down. See?
I guess. YouTube.
It's so weird.
All right, so just tell me if you can hear me now. Can you hear me? Just tell me, can you hear me, guys? I'm hearing, great. All right, so I'm going to be at both places at the same time. I'm going to try to stop what is happening over here. But I still want to see the things that are being said here. All right, and then I'll have to go now to the other link to fix some stuff. But I really want to see what you guys are saying, what you were saying over here. I've seen some stuff that, in my opinion, they are not so accurate. So for that reason, I really want to, I'm not going to kill that. I'm coming right back over there, but I'm going in to fix the setting on this so that persons can find it. Just give me a second. I don't know what's happening with YouTube, but, well, I've been having a headache all day. All right, so I hope that everybody can find me. And what are you doing over there? Alright, so go to live. Okay, great. This is the live. I'm going to go in. I'll be with you in a bit, guys. You could use it for today and tomorrow. Look at that. Yeah. Yeah. CSEC HSB March and Mr. Wilson.
Take it out your mouth. Leave it alone. It's very serious.
Anna, you're being a naughty boy. Yes, you're being a naughty boy. Stop that. Why are you doing that? Huh? Why are you doing that? You think that's cute? All right, you can stop now. All right, come. You're seeking pity now. After you commit your crime, you don't want to do the time. All right, no mind. Hush. All right. All right, great. All right, I am waiting on the system to set something, and then I'll be there. All right, great. So let me see. Okay, so we are here, and I am going to just slow down the talking for me a little bit, right? So I can address the concerns and start the class. Because I think I have some of the answers, probably not all, but I think I have some of the answers. So I'm going to go to the first live, and I'm going to be addressing a number of stuff. All right, I might be old, but I remember a lot of things. So the first question was, biotest was manageable. I think it was. I don't even think. I know it was because I have the paper. Sniper, yeah, it was good. Then Romario Grant said something here. Was it Romario Grant? Romario Grant, the only way you could have said that is that if you are not a part of this channel, because I'm sure we did a past paper with some lizard that actually went somewhere, and we spoke about the finch on the Galapagos Island. We spoke about the length of the giraffe neck. So I don't know why you wouldn't know that unless you came to the class late, but we did address that in a past paper. Then if we were supposed to look at, oh, I see somebody laughing. Wait, so did they have the actual topics mentioned? The exam paper was way too easy for anybody at all to fail. There were just about 3 questions on the paper, just about 3 that students should have a little challenge with. So this time, CXC delivered on their promise. I mean, if I show you some things in on the paper, I have the paper. I'm going to work the paper and send it out. It's just that I'll tell you why the paper is not up as yet. But I have the paper. Here's the paper. Then wait. So yes, they delivered on their promise and more than their promise. I think CXC did an awesome job with this paper. Owing to the pandemic, I give them about a 9. A 9. I think Sharon, how much you give them for this paper? Well, at 9. All right. Owing to the fact that something might be there that I did not see, but I'm giving them a 9 out of 10 for this paper now. Right? At this point, don't trust CXC. At this point, I am trusting CXC. Oh my God, CXC be playing games? No, CXC played no games, and because CXC played no games. So let me continue down this line. Oh my God, oh CXC playing games. They have me learning HSB topics like real hard. We are going to address that tonight. Me nah fret about Wednesday. There's no need to fret. I think I am scared. No reason to be scared. I don't feel ready. That's fine. That's normal. HSB is easy. I don't think that HSB is as easy as biology. Personally, as a teacher, that is what I think because the HSB syllabus is way bigger than the biology syllabus, and too many diseases that you are not accustomed to in real life for you to look at. So that is my personal take. Good evening, everyone. Yes, HSB easy, but I have to remember everything. All right, that's a Jamaican. CXC mark for spelling. No, CXC for science, they don't mark for spelling. Once a word pretty much pronounced, the spelling is pretty much pronounced like the word, and that's why we're not doing GC. For real, like say everyone in this syllabus I go come. Oh, so they won't put everything on it. Biotest was easy. The biotest was good. I tried to revise everything on the syllabus. There was no need to do that. This morning we had the icing, our live that we call the icing, and most of what we covered this morning was on the paper. You know why? I went for the broad topics and prepared the student this morning going into the exam on the broad topic. Anybody who came to my 5 o'clock live this morning will tell you that I was almost spot on with the exam this morning because I was using the broad topic. I tried to revise everything. You should not revise everything because the syllabus told you that everything, the broad topics told you that everything will not be on the exam. All right. So you might think that I am defending CXC, but I'm not defending CXC. I'm defending morality. I'm defending the point that persons are trying to scare other students when they did not prepare for their exams. So I don't want persons to be scaring others because they fail to prepare or fail to follow the protocol. CSEC Biology, the cover page, we brought all the updates that CXC sent. And while we brought these updates, we were working to assist students in doing the exam. So I think it's a little disingenuous for persons to come here to really pull the sword on CXC. CXC delivered, CXC delivered, and from us here at CXC Biology TCP, we are giving CXC a 9 for the delivery. I am surprised that it was the topic they gave us came on the test. I am not surprised, McFarlane. I'm happy there's one person here that can tell the truth. All right. No sound. Yes, I realize that. So pretty much at a glance, those are some of the stuff that I saw, and I wasn't comfortable with the position. Sir, I'm guessing you can end the stream. Okay. All right. My finger up. Okay, great. So that was that over there. So I can sort of kill this live now, and I'm coming over here to look at what you have here. I did ask for you to sort of end the talking a little bit so that I can do a vet of what is happening here to see what we need to deliver on today and what are some of the misconceptions that we need to address. First, I must say good evening. I'm not too sure what's happening with YouTube, but I don't know. OBS I was using, not OBS, I was using StreamYard with the biology kids, and StreamYard wrote me to say that I have exhausted the free version with the bio kids. In a matter of fact, they told me that I'm among 5% of the world's population who stream as much as I do. So you understand that this is not something that I make a revenue from. So I really can't pay to pretty much run a live again with the HSB student. I'm sorry, and my system cannot carry OBS. My system can't carry any software that I will have to download. So for that reason, I will not be having a shared paper session, so to speak. But nonetheless, I'm going to prepare you a little different than I did prepare the bio students. Trust me, you're going to like it. So just stop typing a little bit for me and let me go through, and then I'll tell you what the lineup is. We are expected to be here until about 10 o'clock. That is usually the night class. Okay, here now. Just tell me to close it and click. Click a new link. Oh, yes, we did a new link. Okay, a new link. Oh, all right, great. So, not much said here. All right, good evening, and welcome to Mr. Wilson Live from CSEC Biology TCP and from the TCP Academy. We're preparing students for CXC. We have been here for over a year now, and we have been doing this like 3 times per week. And this is pretty much the final journey for this year. We were fortunate to have had the responsibility to prepare students from across the Caribbean for the biology exams today. And we are very, very proud, and we're really comforted by the fact that what we covered is pretty much what was on the exam. And forgive me, Anna is usually here sometime. For those persons who don't know, he's usually with me sometime. He's my assistant teacher. He's saying hi. All right, great. So the CXC did deliver on the paper, and we are now looking at HSB as to what will happen for HSB. Now, I did promise sometime, if you observe the thumbnail, it has a question sign on it, just to ask if we are going to be doing, nice boy, just to ask if we are going to be doing past papers tonight. The truth is, we're not going to be doing past paper tonight. We are going to be doing better than past paper tonight. Why not past paper? The syllabus is very, very wide, and so too will be the past paper. For tonight's class, you are going to need to have your textbook, one, because I have no StreamYard to show you anything. You are going to need your notebook, and if possible and you have a syllabus, you are going to need it. All that you are going to do between today and tomorrow morning is to look at the topics that will be on your exam. So I'm going to start the show by going through the topics that will come on your exam. I'm going to run through them quickly, and then I am going to teach the topics. Now, I don't have a script for the show, so I will be glancing every now and then in a textbook because I'm not going to be using the syllabus as such, or I might be going from syllabus to book to the screen. But nonetheless, you might not know that because I pretty much have a good coverage of what I'm doing. So pretty much that's what we're going to do. If you are very good at what you do, the class between today and Wednesday morning should help you to pass the exam. So without further ado, I have a past paper here, and on the back of this past paper, I would have written the broad topics. And instead of giving you the icing on the cake Wednesday morning, I'm giving you the icing on the cake Monday evening. Why? I want when you prepare for the exam from now onwards, you are studying only what you need to study for paper 2. So we're going to start by looking at what we should do. Why am I here? While I was preparing the students for biology, there were some persons who knew little or nothing at all, who kept saying in the chat, this is not coming on the exam and that is not coming on the exam. And I'm like, if they ask you to cover that, then this is a part of it. So said, so done. For those persons who have been following, I would have told the students that the nephron would have been on the exam. See there? Excretion. But while I prepared the biology student, there were persons here who were telling us that excretion is not coming. No, I am not going to allow that to happen. I'm not going to be running in any quarrel with anyone. All right? What we're going to do, I am going to be looking at the broad topics here. I'm going to be looking at the syllabus, and I'm going to comb through the syllabus and tell you exactly where the exam will be coming from. As we go through about tomorrow night, I will narrow in on topics or things that must be on the exam. All right? Great. Like I could have told you that genetics is going to be on the exam. The ABO blood group come. I don't know why persons weren't able to do it because I told you that I have a genetics playlist on my channel and it has all that. All right? So persons have to take responsibility for the negligence. Persons have to take responsibility for not being able to do what they should have done because we did what we should have done. All right? Great. So with that said, you mind going down now? Yes, no? Oh, you're not going. Okay, great. So, all right, just sit down then. All right, so you're going down. Right. What are you doing? Make up your mind. All right, great. So let's begin. So CXC said they send up 6 topics that are going to be on the syllabus. That's going to be on the exam. So I'm going to tell you the topics now, and I'm going to tell you all that you are expected to cover for the exam. After which, we're going to start the class, and we're going up to 10 o'clock tonight for those persons who are interested. So I'm going to go from the start of a syllabus to the end of the syllabus, and the syllabus can be downloaded right now from Google if you need a copy. Or probably you can go to the CXC website and get a copy of the syllabus. All right, so great. So we are ready. The syllabus is pretty much divided into sections. Everything in section A could be on the exam because CXC said here in bullet number 1, living organisms and the environment. So section A is the first part of the syllabus. We are going to be going through that in a cram class tonight. So you are going to need to have your book because it doesn't mean that I'm going to be giving notes. It means that I'll be upping over this and upping over that and all of that. So everything in section A, we have to use to prepare for the exam. Have you written that down? Somebody write it in the chat for me because I really don't want persons to be studying what they shouldn't study. So everything in section A of the syllabus, and it is titled Living Organism and the Environment, everything there we are going to touch. Don't worry yourself. You soon know what are the other parts under everything. All right? Please be reminded to like, share, and subscribe, and call your friends to come on over. All right? Subscribe only if you like what we do here. If you don't like what we do, you don't have to subscribe. All right? So we want really organic persons who like what we do and really want to share what we do. Like Amoy likes what we do. And I like Amoy likes what happens here. And I know some other persons. I'll call names as I go along. All right, great. So everything in section A, living organism and the environment. We'll soon break that down. Let's go back to the syllabus. So I'm using the syllabus to tell you now and nothing else. So after section A, what comes next is life process. So we are in section B of the syllabus. Now, nutrition will not be on the exam based on what CXC sent. So that is number 1 under section A. So we don't need to study about the teeth and digestion and all of that for the paper 2. All right, great. So let us go down and let me tell you what else is going to be there. Respiration will not be on the exam based on what CXC sent. So 1 and 2 will not be there. Let's continue. That is why I didn't want to do any past paper, because we're going to be doing things we don't need to do. Circulatory system will not be there. We don't need to know about the blood and lymphatic system and all of that. That won't be there. Let's move on down. We are at, wow, this will be there. The skeletal system will be there. So type it in the chat there for me. So after section A, we are going to go to section B, and in section B, we are going to be looking at the skeletal system. That's a big thing. We are going to do that probably tonight. We have up to 10 o'clock. Now, this is the part that we have to pay much attention to, because it has some amount of depth. The excretory system and homeostasis will be there, which means that we'll have to look at the kidney, the nephron, the skin, negative feedback mechanism, positive feedback mechanism. We'll have to look at all of that. Then after the excretory system and homeostasis, then we have to look at coordination and control is not there. So we don't need to study about the brain and the nerves and all of that. That won't be on the exam. Then after that, the next thing I'm going through the syllabus is a reproductive system. I'm having this jolting headache. Will be the reproductive system. That is bullet number 7. Then after bullet number 7, our next topic that we're supposed to cover is going to be heredity and variation. That is section C of the syllabus. So we did point out some things from section B that you are supposed to cover. Only 3 topics from section B that we're going to be covering: skeletal system, excretion, and homeostasis. And we're supposed to link it to chronic lifestyle diseases. We are supposed to do that, so you understand that we have to put a little thing more than you are going to find there in it. So we are going to tell you what needs to be added to it. We're supposed to look at reproduction, I said, from section B. And now we are at section C, heredity and variation. We are supposed to look at everything in heredity and variation. Then after heredity and variation, section D, this is a little tricky. Section D, which says disease and the impact on human, is not on the exam paper. However, there are aspects of section D on the exam paper. So you're not going to be having a big thing on the paper. So let me show you how it will be on the exam paper. It will be on the exam paper because section E of the syllabus, which speaks to the impact of health practice on the environment. So the impact of health practice on the environment, we would have spoken about sewage, we would have spoken about the various pollution. That is the very last part of the syllabus, water treatment and all those stuff. That, of course, will be on the syllabus. So we're having a crash course tonight. It means, therefore, that you would have studied and you can follow the pace that I'll be going. For those persons who know me, when I pick up speed, I can go really fast. So it says here, for section E, we are supposed to link to infectious disease. So all those waterborne diseases that we spoke about, the housefly and the disease that are associated with it, for those persons who have been a part of my life, all those slides we need to go back to. Now, with that said, I am going to be upping on into the class so that the time will be wisely spent. So there we are having a number of persons, giving those persons the topic to study. And I want you to take this serious. Why we're giving you these topics is because Mr. Wilson will not be able to cover all of them. I'll be able to just jump over some of them, and then you will have to go and do the study on them. All right, but we don't want it to be studying all over the place. It makes no sense for me to pop on a past paper now, because if I give you a past paper, we might be looking at things that we don't need to look at, right? And I don't want that. All right, great. With that said, I am going to be upping on into living organism and the environment. I'm going to be coming from my head for the greater part, but you might see me pause every now and then, because I'm not going to be telling you anything that I think should be the fact. If I don't know, I'm going to check or ask you to check. That's Mr. Wilson. Mr. Wilson has never been the keeper of knowledge, and Mr. Wilson is willing to accept correction at any stage of the game. All right, so here we go. I'm going to go to the syllabus. I might not be seeing the screen. So if you're typing and I'm not seeing, don't be mad at me. Is that I am not looking at, even though I'm looking at the screen, I'm looking pretty much at the syllabus, because that is what I'm going to use to get you ready for the exam. So we need to know the difference between cells. We need to know the difference between the type of cells. So I am ready to start, and I don't have a physical syllabus, so I'm looking at a soft copy. So the first part of your syllabus looks at characteristics of living things. Characteristics of living things, quickly type them in the chat for me. What are the characteristics of living things? And it said that you're supposed to be able to describe the characteristic of living things. Yes, we have a video that we covered on our channel on characteristic of living things, which is doing really, really well. All right, so you want to type them just type the characteristic of living things one time and post it. Don't type them one. Just type them one time and post it. All right, one time and post it. Don't type them one. It's going to take up too much space and too much time. After we would have looked at the characteristic of living things, the other thing we are going to look at on the syllabus, we are supposed to compare the structure of unspecialized cells for plant and animal cells and selected microbes. Now, it's important to note that the microbes that we cover for biology is not the same microbes that we cover for HSB. So we are going to look at the basic structure of an unspecialized cell. And when we talk about an unspecialized cell, we are just talking about the general plant and animal cell. Now that we have done that exercise that I gave you, I am going to be giving you another exercise. So just let me, I'm sorry, I can't just pause the screen and say. So remember, to remember the characteristic of living things, it's simply Mrs. Gren, a reminder. All right, but Mrs. Gren is the safer thing to use. So we have movement, Mrs. Gren. Let me see if I can remember them. Movement, irritability or sensitivity, that is MS. Growth and development, that is G. Reproduction, GRE. Excretion, nutrition. And did I say respiration? That should have been the missus. So it's MRS, movement, respiration, sensitivity. And then we have growth and development. Then we have reproduction. And then we have nutrition. So when you get to that exam, there are many names I've never seen this. Miss Inga or Inger. All right. So when you get to that exam, if you don't remember these, though these might be basic for a lot of persons, just remember to write down your Mrs. Gren, and I'm seeing somebody there writing some other thing. You could type the remind. Oh, we're seeing it. There are so many. Grimner. So Grimner. So I'm seeing a lot on the screen. So these are memory aids. You have Mr. Grens as well. Mr. Grens. So Mr. Grens, Grens. That's another memory aid that you could use for characteristic of living things. Very well. That is out of the way. We're past that. So we're moving on now. I want you to write one time all the parts of the animal cell. Remember, for HSB, it is not a part of the biology. You need to list when you are talking about your cells, the ribosome and the endoplasmic reticulum. So I want you to write just one time all the parts on your syllabus for the animal cell. I'm going to make it a little spicier. I want the girls to write all the parts for the animal cell, as it were, for the CSEC exam. And I want the males in the room to write all the parts for the plant cell. So you're going to write everything one time and pause. Everything one time and pause. All right? So the first person to come up with it right, then that team will now give us a distinction between the plant and animal cell. So let me see who are the persons who are doing it. Okay, great. So I am seeing Anissa Small. Is it male or female? Tell me in the chat and I'll tell you what will happen. So we have Anissa Small or Anis. This is not everything. I'm seeing ribosomes, I'm seeing endoplasmic reticulum, I'm seeing small, that's not everything, I'm sorry. All right, animal cell, I'm seeing cytoplasm, I'm seeing endoplasmic reticulum, I'm seeing nucleus, ribosomes, cell membrane, mitochondria. Watson, that is not everything. I think this is Shelley. Cell wall, cell membrane, nucleus, ribosome, cytoplasm. That is not everything. Let me go again. Nucleus. Okay. Okay, great. So Campbell, I'm looking for the glycogen granule. Guys, you are not remembering the glycogen granule. That is how the animal cell stores starch as glycogen. So you cannot allow yourself not to remember that. Cell membrane, cytoplasm, nucleus, small vacuole, mitochondria, glycogen granule, ribosomes, endoplasmic reticulum. You must be able to name that. All right? So Amoy, you're a boy. Plant cell. So for the plant cell, cell membrane, ribosome, cytoplasm, nucleus, rough endoplasmic. You can just say endoplasmic reticulum. You don't have to go smooth or rough. Smooth endoplasmic reticulum, mitochondria. Guess what? Amoy, your plant cell don't have any cell wall. I wonder why. You can't afford to forget that. You can't afford to forget that. So I am not using anything, and I remember, and I am getting old. All right? So great. I'm seeing a lot of things here. So I'm going to try to see how best. So stop typing a bit for me. Stop typing a bit for me so that I can make the correction and move on. We don't want to stay on one thing too long. All right? So just stop typing a bit for me. So if we are looking at the cell, the animal cell, let us be reminded. Let us be reminded. Please listen now. But it said plant cell. I think you wrote plant cell. Amoy. I think it said plant cell. All right, just stop typing a bit for me, please. Let me correct this and move on very quickly. So the animal cell, it has a cell membrane, but no cell wall. The plant cell has a cell wall and cell membrane. The animal cell has small vacuoles, while the plant cell has a large vacuole. They both have cytoplasm. The animal cell has glycogen granule. The plant cell has starch granule. They both have nucleus. The plant cell has a chloroplast. The animal cell does not have a chloroplast. Did I say the plant cell stores starch granules and the animal cell glycogen granule? What am I missing? They both have mitochondria. They both have endoplasmic reticulum, and they both have the ribosome stuff, ribosome for protein synthesis. All right. I hope I haven't missed anything. I really wanted to look at the screen to make sure that I'm not missing anything. All right, guys, please, when I am teaching, I am one of those discipline freak, right? If you are typing all over the place, you are distracting persons, right? We want you to keep your conversation around what we are covering when we are covering it, all right? We don't want persons to be going left, right, and you are distracting me because I'm looking at food chain. We're not doing food chain now. We're going to get to food chain, all right? Great. So let's keep it that way. We want to make sure that we are focused and we are all getting there pretty much almost at the same pace. All right, great. Now, other than knowing the parts of the cells, the generalized cell, you are supposed to know the difference between the plant and the animal cell. You are also supposed to know the function of the organelles. So all these parts that we have just made mention of, the cell membrane and the mitochondria and all of those stuff, these are organelles of the cell, and you need to know the function. One of the things that you need to remember about the cell is that the mitochondria and the chloroplast, they are somewhat different. Now, the mitochondria will have what we call invagination. And I am out of paper. My 500 pack, for those who know me, I don't have any 500 pack. Okay, I probably can write on the back of this. So if you are looking at the, I'm just going to try to draw this here. If you are looking at the mitochondria, you'll see it having something. I hope this is not too small. You can see that little invagination inside. Good. While if you are, that's the mitochondria. While if you are looking at the chloroplast, it tend to have stocks of what we call cristae, something looking more like that, right? Something more looking like that. So the one at the top is the mitochondria, and the one at the bottom there are stocks of cristae. That is going to be the chloroplast. Now, you might be given a micrograph, that is a picture taken under a microscope, to identify it. You must be able to do that. All right? So that's it for the plant and animal cell. Knowing the difference, be able to identify the parts. It said the basic structure of unspecialized plant and animal cell. And I think it says somewhere here exactly the parts that we're supposed to know. As soon as I get there, I will just go back through that. Then we're supposed to know the difference between microbes. Now, the specific microbes that we should be looking at are viruses. You can say viruses. Bacteria, that is plural. And the singular for bacteria is bacterium. And we're supposed to look at fungus, which is singular, which is plural. Fungi or fungus. Tell me in the chat which is singular, which is plural. I tend to learn from you as well. I am as dumb as a dumb. All right, great. So when we look at these three particular organisms, they could appear on your exam, anyone. So you need to just flip your book to look at them. Now, a virus is not a real organism, and you could see a virus appearing as a phage, something looking like a spaceship, or you could see something on the paper looking pretty much like that you have been seeing for the coronavirus. All right. Now, the viruses are virus that really can't exist outside of a host. They are always inside of a host. You need to be able to identify them. I don't know that you are ever asked to draw anything in HSB, but you might be able, you might be asked to identify a particular part of the virus, or a particular part of the bacteria, or a particular part of the fungi. You must also be able to distinguish among these microbes. Now, please be reminded that the bacteria there, they don't have any membrane-bound organelle within them. They are actually what we refer to as prokaryotic organism. They tend to have a flagella or flagellum, and of course, they also have a cell wall, pretty much like the plant cell. So you want to make sure that you are looking at your textbook now at the bacteria or bacterium. You might be asked to identify or to tell the difference between the virus, bacteria, and fungi. Now, it's very important to note that while viruses, they are pretty much straight disease-causing agent, the bacteria or bacterium, they are pretty much in between. They could be destructive, and if you go to my video that covers these pathogens, you'll see it. They could pretty much cause a whole lot of problem, as well as they could be of value. So you'd know that they are useful bacteria. If we even think, the quick one I can think about is like the rhizobium, but of course, bacteria is of course used in production of a couple of stuff, right? When you do respiration, you'll see that. Then you have to think now about the fungi. Now, the fungi pretty much, it doesn't carry out photosynthesis, right? The fungi feeds on what we refer to as hyphae, and the group of hyphae will refer to it as mycelium. Now, mycelium, what happens is that the fungi will pretty much send out things looking like roots, and it will pretty much carry enzymes that will digest whatever it wants to consume. Now that we have spoken about virus, bacteria, and fungi, it's important that we now add to the mix that both bacteria and fungi, behave yourself, man. Both bacteria and fungi, they are part of what we refer to as saprophytic organism. Now, saprophytic organism, to be specific, bacteria and fungi, they are both referred to as decomposers. Now, you might see on your exam paper, as it was on the exam paper today, you might see on your exam paper to tell the role of decomposers. Now, the role of decomposer is that they tend to cycle nutrient in the environment. So they assist with decomposition and to release trapped nutrient in dead and decaying organism. That is pretty much the function. Now, when we talk about detritivore, or when we talk about saprophytes, we are now going to tie this into nutrition. There are three types of nutrition. Autotrophic nutrition, that is carried out by plants, organisms that make their own food. Then there is heterotrophic nutrition, that is carried out by organisms that are depending on other organisms for food. So if you take, for example, plants, what they do is to use simple inorganic substances to make food. And then the heterotrophic organism, they rely on that plant to get that food. Sorry, the heterotrophic organism, they rely on the plant to get the food. And then there are different types of heterotrophic organisms. Then there is the saprophytic nutrition, which rely on the dead and decaying organisms in order to have food. Now, on the paper today, as I said in the live this morning, if you are using a food web and you remove an organism, what will naturally happen is that the organism below might increase in numbers. The organism above might die, migrate, or feed on some other organism. So in the paper today, they had asked, which they can ask you in HSB, why is it that we have less organism at the top of the food web? It is that the nutrient, the amount of nutrient being moved from producer up through the food chain or food web, it reduces, so there's not enough energy to sustain the organism at the top. I have said a lot, and I will come back to that. All right. So we looked at decomposers, as it were, with the cells. I tied it up into cells nicely, and we're going to be moving on to something else now. So please be able to identify bacteria, virus, and of course, fungus. As it were, for biology, they looked at protozoa, and they did not look at, I don't remember what they didn't look at. They didn't look at, I think, was it virus? I don't remember. I really don't remember. I'm sorry. That is not even important now. All right, great. So we would have passed specific objective 1, specific objective 2. I would have outlined all that you are supposed to know. At specific objective 3, they would have pretty much told us what is it that we need to be able to label when it comes to the generalized cells. What is it that we need to label? We need to be able to identify the cell wall, the cell membrane, the nucleus, the ribosome, the cytoplasm, the mitochondria, the vacuole, the chloroplast, endoplasmic reticulum. So one of the things when you are studying, you want to use number code to remember these things. So cell wall, cell membrane, nucleus, ribosomes, cytoplasm, mitochondria, vacuole, endoplasmic reticulum. So there are 8 organelles that are supposed to be you are supposed to remember. Remember, please be reminded that not all these organelles are found in both plant and animal cell, and you must be able to tell the difference. That is specific objective 3. Specific objective 4, identify selected cells which make up the human body. Identify selected cells that make up the human body. Now, you don't need to just go about and just look for any cells because the cells are named in the syllabus that you should know. Now, the syllabus, the cells that you are supposed to know diagrams and features of the following. So you need to know what they look like. Remember today on the paper, they asked them to draw the white blood cell and the red blood cell, and they were supposed to label them. So they should be able to identify the nucleus, the biconcave, this, that type of a thing. So you are supposed to be able to label these, should you see them on the paper. The epithelial cell. Now the epithelial cell tend to form the covering of organs. All right, epithelial cell, they form the covering. You are supposed to be able to identify and label the sperm cell, the acrosome, the tail of the sperm. You need to be able to identify and label that. You need to be able to identify and label the egg or the ovum with that large nucleus. You should be able to identify nerve cells. Now, there are 3 types of nerve cells that you are supposed to be able to identify. 3 types of nerve cells. Yeah, it might be an overstudy. I think so, but that's fine. You are supposed to be able to identify 3 types of nerve cell. There is a small nerve cell that is called the intermediate relay or connector neuron. That's a small one. Then there is a long one with a cell body somewhere in the middle there. I think that one is called the sensory neuron. And that is pretty much taking information from the receptor upward. And then there is the motor neuron that has the cell body at the end of this long process, and it is pretty much carrying back things to the effector. So those are the 3 that you should know. Remember, one has a cell body somewhere in the middle there. Another is just a little small thing with some sprung like a circle. Then there is another that has the cell body pretty much at the end of a long process. Don't we have to draw in HSB? No, we don't draw in HSB. I've never seen that. What we do is label, not even to label, like to identify the labeled part. So they might label somewhere A, B, C, or D, and then you're asked to pretty much identify A, B, C, or D. I'm sorry. I don't know who it is. I'm sorry. It's a Jamaican number. I don't know. All right, good. So let us move on. That breaks my thought just now. So we looked at that. So the other thing that you need to look at is your muscle cells. All right, remember that you have different types of muscle cells. You have different types of muscle cells. You have the cardiac muscle. You have the striated muscle. And what is the other muscle? That one with the bands and the nucleus? That one. So they are really about 3 dear type of muscle. They are somewhere in the front of the book. Forgive me, my brain is not always ticking as it should. So there are about 3 types of muscle cell there. You want to be able to identify them. I don't think you need to write anything on them. When we get to the skeletal system, we'll talk about that. So here we have some stuff. Not everything is in your textbook either, because I'm looking at the textbook and not everything is there. Dark and light band. Yes, that one. Yes, and then you have this one, the smooth curve, nice one, smooth muscle, right? Very well. Cardiac muscle, that's the one in the heart, and then you have the striated muscle, right? Smooth muscle, you have the striated muscle, you have the cardiac muscle. So you need to be able to identify muscle. Let me just up on back over here and tell you. So what you are doing now, you are going through your notebook and look at the things that you're not able to identify, and you are now going to be doing that. You are supposed to be able to explain the importance of cell specialization. Now, cell specialization or differentiation, it speaks to a cell becoming specially adapted to carry out a specific function. So we know that it is not the nerve cell that gets a woman pregnant. It is the sperm cell. So cell differentiation or cell specialization, all that it speaks to is the ability or a cell becoming specific, or a cell taking on this specific function. So you know that, for example, when we look at specialized cell, we could talk about the nerve cell. We could talk about the blood cell and the different types of blood cell. We could talk about the epithelial cell. We could talk about the muscle cell, and the number of cells go on and on and on and on. So we talk about cell differentiation. All that it means is that cells now are specialized to carry out specific activity. All right, great. So we move on to the other thing that was cell specialization, and the syllabus, what it says, cell specialization. So we're moving on down and we are going very quickly, but I'm sure that after this class, you're going to know exactly what you don't know and what you should have. We're supposed to, in relation to cells, to the organism as a role. Relation of cells to organism as a whole. All right, great. Let me just treat with that. So it says in relation to cells and organism as a whole. That is now speaking about the hierarchical development. And walk with me on this journey very quickly. So the cells are made up of organelles, and then a group of organelles will of course make up a cell. Then a group of cells will make up a cell will make up tissue, and then group of tissue will make up organs, and then group of organs will make up a system or organ system, and then a group of organ system will make up a organism. All right, great. And then a group of organism will make up a. All right, great. I like what's happening. All right, just hold one bit. I think I upped over something. I don't remember if it is on your syllabus, but I think you need to know. So when we get to organism, we have a new word being formed. The word species. Species always end with a S. So when you get to organism is the same thing as a species. All right. Now, a group of organism or a group of the same species will make a population. All right. A group of same organism or of the same species will make a population. A group of, so we talk about population, we're talking like about a flock of bird, we're talking about like a pride of lion, we're talking about an orchard of orange, we're talking about an orchard of breadfruit, and then a group now of different population. So a coconut tree, a mango tree, a breadfruit tree, a ackee tree, a soursop tree, human, a dog, a cat, all of that comes together to make a community. And then this community interacting with the abiotic component, the wind, the water, all of that makes up an ecosystem. So that's the hierarchical thing in relation there. You must have cells in order to have tissue. You must have tissue in order to have organ, and you must have organ in order to have system. All right, great. Now, there are different types of tissues. There are what we refer to as muscle tissue, nerve tissue, connective tissue, epithelial tissue. So those are pretty much the general 4 types of tissue. You have connective tissue, of which the blood is a connective tissue, the bone is a connective tissue, the cheek cell is an epithelial tissue. Then we talk about the nerve cell, and we talk about the different muscle. Talk about the nerve as a tissue. All right, great. So we would have done that in pretty much the hierarchical system. So we looked at that very quickly. 4 types of tissue. Wow, we punched that in nicely and we are moving on. All right, great. I know that you're not doing much talking now, but forgive me. I think these are like the first part of the syllabus, and you should be good at that. Now we are supposed to be able to distinguish between osmosis and diffusion. We should be able to distinguish between osmosis and diffusion. Now, osmosis, what is osmosis? We have, for the persons who were on the live last night, you would have seen that. What's osmosis? Now, to write two definitions is not to distinguish, all right? When you are distinguishing, you are going to write one definition and then you are going to separate it by while. All right, so just type in the chat for me. I want some persons to type what is osmosis. So you're going to say osmosis is whatever it is. And then other persons are going to say diffusion, it's what it is. All right, osmosis is the movement of water particles through a selectively permeable membrane. Great. So what is diffusion? Now remember that in osmosis, it is water that is moving. In osmosis, it is water that is moving, and this is, of course, going through a selectively permeable membrane, allowing mineral to pass through a semi-permeable. I don't know that we're still using semi-permeable. Just let me check the syllabus. I was told sometime that we're using selectively, and I could explain why not semi-permeable, because if semi-permeable, some things can go through, but with selectively permeable, it will allow what it wants to go through. So not because water can pass through the membrane, the cell will determine when the water passes through. And that is why it is selectively permeable and not so much semi-permeable. I know that a lot of textbooks, they are still carrying the semi-permeable concept, but I think we would have long passed that. Osmosis is the movement of water molecule through a partially permeable membrane. Oh, we're going so fast now. A partially permeable membrane from a solution containing a lot of water molecules to, all right, so the water molecule is moving from a place of high concentration where there's more water molecule to a place where there's less water molecule that we call a concentration gradient. All right, so we could say that it is moving down a concentration gradient. Now, diffusion, on the other hand, is a movement of molecule or movement of particles from a place of high concentration to a place of low concentration. With diffusion, we are looking at all types of particles, and that is why osmosis is said to be a special type of diffusion. But always remember, there are two things that are key about osmosis, is that osmosis is going through a selectively permeable membrane, and osmosis is the movement of water. So tell me in the chat, we have some syrup. We have some syrup in a tissue funnel and the syrup is moving. Is that diffusion or osmosis? McIntyre Wilson, I saw your message. I think I responded. All right, King, did I see something? So you see what's happening there? A lot of persons are not listening. And that is why when I'm talking, I'd love for persons to pay attention. If you have a question to answer, I don't know why we have asthma regulation. I don't know how that fits into the mix. I'm sorry. All right, great. Remember what we said about osmosis? It's the movement of water, not the movement of liquid. We said that osmosis is the movement of water. Syrup is not water. Syrup is a liquid. So it is diffusion. Remember, osmosis is the movement of water particles. Osmosis is the movement of water particles, not liquid. All right? Not liquid. So if it's iodine or any other type of liquid, it is diffusion. Osmosis is the movement of water particles. Great. All right, so we move back into the syllabus, and I think most person would have had that. I don't know, I'm seeing a footnote here for diffusion. I'm not too sure why it is there. So you are going to be asked to distinguish between. So you are going to give the definition for one, and then you are going to say while and give the definition for the other. Then it continues to say that we are supposed to explain the importance of osmosis, diffusion, and active transport in living system. For those persons who were at the live last night, we did this. We looked at the importance. We looked at 4 importance last night for diffusion, and probably we did look at 4 importance for osmosis. It continued to say here that movement of substances across cell membrane. Osmosis is a special type of diffusion. Cell membrane is a partially permeable membrane. Well, the syllabus is now saying partially permeable. Movement of water molecule from cell to cell These are things that we should look at. We're supposed to give examples of osmosis occurring in plants and animals. When we are answering this question, the question that says that we are supposed to explain the importance of osmosis and diffusion, it is saying that we are supposed to look at the whole idea of osmosis as a special type of diffusion, the importance of that. So we could look at the movement of water within the organism. All right. And then it says that we're going to, and when we talk about water in the organism, we have to also look at water as it is plant takes water from the soil through osmosis. There we want to pay attention to that. Not just human, but osmosis in plant, as it were, for you. Then we look at the cell membrane, which is, of course, partially or selectively permeable, that is allowing substance to pass through, water to pass through, that type of a thing. So the cell membrane there would have served as the selectively permeable membrane, just like a tissue fluid. So when we talk about the cell membrane, it is the cell membrane that is selectively permeable, while the cell wall is freely permeable. Now, the movement of water from cell to cell, yes, that is needed for, that is pretty much another thing in osmosis. We're supposed to give examples of osmosis occurring in plants and animals. And we spoke about osmosis as it moved from the soil, water moved from the soil into the plant. Now, we're supposed to talk about diffusion of gases, and we could talk about diffusion of gases as it were for photosynthesis, where the carbon dioxide is diffusing into the leaf and oxygen diffusing out. There, that is through the stomata, and that is pretty much gaseous exchange. We could also talk about gaseous exchange as it occurs in the lungs. Now, we could also talk about the oxygen and the carbon dioxide exchanging there in the blood cell, as there is a concentration gradient between the cell and pretty much the blood vessel. Now, another thing we're supposed to look at is the experiment. Oh, there's an experimental activity to demonstrate osmosis and diffusion in living cells. So let me just come on over. A lot of teachers like to tell you that there is no lab for HSB. It is not true. So the labs are, let me give you some labs that I can remember very quickly. The lab for diffusion and osmosis for your diffusion, for your osmosis exam, in most cases, they are using like a potato. And let me tell you what will happen. Jot this down if you did not do in the lab this year. If you are using a potato strip and it is placed into water, that is what we refer to as a hypotonic solution. So what will happen is that there is more salts in the potato strip. So the water will move from the container with the water into the potato strip. So if you had actually cored the potato out, you'd be seeing water inside of the potato. To enhance this or to allow it to happen quickly, we usually core the potato out and put some salt in it that's more concentrated, and the water will pretty much go through the tissue of the plant and be collected there. If you're using like a potato strip for osmosis, what will happen? As the water gets into the potato strip, it will increase in mass, it will become turgid, so it will feel a rough, the potato strip will feel rough as the water is pretty much pressing onto the cell wall there, making it really rigid, tough. Now, if you were supposed to put the same potato strip into some salt water, sugar and water, anything that is concentrated, now this is a hypertonic solution. Now, a hypertonic solution has more solute than solvent, right? So what will happen now is that the water that is in the potato would have moved out of the potato into the solution. All right. So we spoke about that last night when we were preparing for biology, where we spoke about hypertonic and hypotonic solution. There's also isotonic solution, being that most of you guys are going to be doing nursing. In the isotonic solution, what happens is that the solution is pretty much at a similar concentration as to that of the potato strip, or as it were with the case of the IV fluid and the IV fluid and your body concentration, it is somewhat similar. All right. Great. So that there, we looked at osmosis and how it could be tested. Just try to figure out where is the most concentrated solution, and wherever we have a more concentrated solution, we are going to be having the water moving in that direction. All right. Then there is, yes, flaccid. When we put that potato strip in the salt solution, it will become plasmolized, which means that the cell membrane will leave the cell wall and shrink on the inside. As a result, the thing will become flaccid, bend easily, and it will become a little smooth because it has lost water, so it is not as stiff as it used to be. It just is easily bent. So those are some stuff that we have to look at, and that is the case, as it were, for plant cell. If we're supposed to look at the animal cell, if we were supposed to place the animal cell in water like we did in for the plant cell, the animal cell does not have a cell wall to protect the cell membrane. As such, the plant cell, the animal cell would rupture or burst. All right, there are some other fancy words, but we can stay here. I will spoke about the saltwater fish and the freshwater fish. The concept is the same. The only difference with fish is like the salmon. The salmon is able to move from freshwater to saltwater because it can pass its urine and control the internal environment. So if we put the animal cell in fresh water, which is pretty much a hypotonic solution, then what is going to happen naturally is that's going to take in water and eventually burst. That is why the drip that you take when you go to the hospital is not pure water. That is why you can't take a saltwater fish and put it into fresh water, because what will happen is now a hypotonic solution and the water will move inside of the fish and pretty much cause the cells to burst. Now, if you were supposed to place this animal cell in salt water or anything with more solute, what would have happened is that the water would have moved out of the cell into the hypertonic solution, and then the cell would become shrink, as we would have said, shrink, all right, as the amount of water there in the cytoplasm would have been moved out. All right. So when we are doing the drip, as it were, remember it should be in an isotonic environment. So we spoke about hypertonic, isotonic, and hypotonic. Great. Then we look at diffusion. Now remember, when osmosis is taking place, there must be a gradient. There's a gradient when we have hypo and hypertonic solution. Gradient meaning one higher than the other, so one has more solute than the other. When we're looking at diffusion, diffusion is pretty much a movement of particles from one place to another. So if you spray it on your cologne upstairs, then somebody downstairs is going to smell it. And if you're wearing a good cologne because you're one of those affluent students and you walk through the town, persons will smell that cologne as you pass, as the particles continue to diffuse. Now, if you're passing by a restaurant and you would have smelled what is cooking, that is diffusion. If you have some scented white flower or some scented plant in your house and it's like night and you start smelling something, that is also diffusion. So diffusion is pretty much a movement of particles from a place where there's a lot of the particle to a place where there is less of that particle, or it's moving from a place of high concentration to a place of low concentration. All right, great. So let us look at what else we need to cover there. A lot said. I know a lot of persons are, whoa, I'm overwhelmed. Do it look like I'm saying so much? Trust me, guys, it's not that bad. All right. Then we have active transport, and this is one of the things that most persons don't know or is never covered. Our teachers tell you, go and read on it, and you really don't understand. All right, so it's a movement of ions against a concentration gradient. So always remember that while osmosis and diffusion is going downhill along with the concentration gradient, active transport is going uphill because it's going against the concentration gradient. Now, usually active transport is carrying ions, and it's like sodium ion, that type of a thing. And of course, it is using energy from the cell to get the job done. Most person will tell you that active transport uses energy from the cell, but as to the part where it moves against the gradient, most persons are not remembering that part. So it's like diffusion and osmosis is going that way, active transport is going that way. So it's going up the hill, right? Against the concentration gradient, and it is using energy from the cell. Always remember that. It's using energy from the cell and it's going against the concentration gradient. You need to know the importance of all these three. I would have outlined that, and take a glance at your text as well. Now, when it comes to photosynthesis, you are supposed to be able to define photosynthesis. And there are many definitions for photosynthesis, and some tend to mean the same thing, some tend to be a little different. Now, as it were, for, okay, great, somebody saying passive transport versus active transport. So passive transport refers to diffusion and osmosis. Passive transport refers to diffusion and osmosis, right? And active transport, we spoke about that. It's started by itself. Now, photosynthesis, we have some definitions that are a real misconception. And when you come to Mr. Wilson class, I'm going to clear them up. You understand that when we're walking, we really start down here and we fell and then all of that. So don't blame your primary school teacher to tell you that photosynthesis is the process by which green plants, don't blame them to tell you that thing, all right? Because it's what you could have understood then. So photosynthesis is the process by which plants use simple inorganic substances, which is pretty much water and carbon dioxide, in the presence of sunlight and chlorophyll to make organic compound and pretty much oxygen. Let me slow that down. When photosynthesis has what we call a formula, or equation, some might say, it has three parts. The first part, which is water and carbon dioxide, is called the raw material. I don't want anything about any green plants, and I soon deal with that. The first part of the equation, which is the carbon dioxide and water, is called the raw material. The part over the arrow is called the condition, and that includes sunlight and chlorophyll. Yes, a plant can carry out photosynthesis without the sunlight. What is needed is actually light. Why do we say sunlight? Because it is a school of thought that all light came from the sun. The sun is the ultimate source of energy. So not because we're saying sunlight. If you turn on the light, the plant will carry out photosynthesis. All right, great. So photosynthesis is the process by which green plant use simple inorganics. Why we can't have a green plant? Have you ever seen any plant that is red or the croton or anything like that? Are we supposed to assume that they don't carry out photosynthesis? No, it is not so. For those persons who are from Jamaica who know the plant we call dragon blood, it is called Dracena. I don't remember the botanical name. It is somewhat burgundy in color, or the leaves are burgundy in color. It carries out photosynthesis, right? But plants that are not green, they tend, and this is biology now, plants that are not green, they carry out photosynthesis using what we call accessory pigment. I won't tell you the name of the accessory pigment because you don't need to know them. All right, you don't need to know about accessory pigment. But I just want you to know that this green plant, we don't need to put it there because all plants will carry out photosynthesis. Whether they are CAM plants, C4 plants, C3 plant, they are going to carry it out. You don't need to know about CAM, C3, or C4 plant. You just need to know about plants. All right, great. So we understand the equation now for photosynthesis, that it starts with carbon dioxide and water. Then the sunlight is going to provide the energy. It is not the heat energy from the sun that is providing the energy for photosynthesis. It is the light energy from the sun that provides this energy, while the chlorophyll will trap this energy, like catch it like that, trap this energy, and it uses this energy to split the water molecule into oxygen and hydrogen. We'll soon talk about that part. And then it's going to make glucose that is pretty much converted. On the paper today, they had asked you why glucose is not stored and it is starch that is stored. You want to follow the paper when I publish it and you'll get the answer for that. All right, so great. So we have producing from photosynthesis glucose, and you're supposed to know the fate of the product of photosynthesis. So that is where you should have gotten that answer from when you were supposed to answer that question, but you didn't remember that, right? Great. And we have oxygen that is produced, and the oxygen is given off, and it is used by other organisms for photosynthesis. All right? You don't need to worry. You don't need to worry. Trust me. Not because you hear me talking so much stuff. These things, when you look at the paper, you're going to see something like a summarized version. So I'm just touching little this and little that. All right, so I'm going to go back to photosynthesis. I can remember it. I'm not looking at a book to tell you, I can remember it, and so can you. Trust me. Trust me. So we need to know that definition for photosynthesis. All right, great. Then we're supposed to have simple treatment involving word equation. So you are only supposed to know the worded equation. Biology need to know the other type of equation, and worded equation I just told you. Water plus carbon dioxide, sunlight over chlorophyll with that arrow, glucose and water. That is all you need to know about the equation, and you need to know the parts of the equation: raw material, condition, and product. Then it says here we are supposed to cite the site of photosynthesis. This is now where you have to remember from your plant cell that the organelle in the cell is the chloroplast. And the chloroplast is the site of photosynthesis in the plant. The chloroplast contains chlorophyll, which traps the sunlight, and it is in the chloroplast that photosynthesis is taking place. So when you see the exam is asking you, what's the site of photosynthesis? It's the chloroplast. All right, great. So let us continue. Another thing it says here is the fate of the product of photosynthesis. When it asks us about the fate of the product for photosynthesis, for the first time I feel like going to my book. All right, when it asks for the fate of the product of photosynthesis, it's asking you to tell what happens to the things that are made in photosynthesis. So what happens to the glucose that is made in photosynthesis? It is converted and stored. Now, on the paper today, they did ask, why is it converted and stored? So it might be wise for you to do that, because I'm sure that CXC know that that question is going to mold everyone. If anybody knows, just type it in the chat. I'm just looking at something on the screen. Obviously, this person is not a YouTuber. If you are a YouTuber, when a person has a live, you can't advertise. YouTube will not allow it to show. All right. If so, persons from time to time when I'm having live, there are persons who come on with pornography and all of those stuff. So don't do that. It is sort of unethical, and YouTube is trying to cut out those type of stuff. All right. Great. I know that everybody wants persons to follow them on their channel, but just make yourself great product and wait your turn, and it will happen. All right. I am very, very humble. I mean, yeah. All right. Or if you want to be a part of my live one day, just send me a message and I'll invite you over. It really don't matter what you do on your channel. Whether you do biology or HSB, it doesn't really matter to me. All right. The exposure I will assist persons with. That's fine. So because glucose is soluble in water and will disrupt the osmotic balance of the leaf. Now, oh, who said that? That sounds like music, eh? Great. While starch is not as soluble. Hey, that is what you should have written on your exam today. All right, and you didn't remember. That's definitely the answer. All right, great. So we move on. So remember that the glucose is converted and stored because the glucose is so soluble, so it has to be stored in a form that is not as soluble. All right, great. Another thing about this thing here is that the oxygen is given off, and the oxygen is used for combustion, and it is used for respiration. All right, you could rest your ears a little bit. You are watching CSEC Biology, TCP. I am Mr. Wilson from the TCP Academy. We have an online platform that persons can pay for products there, or pay for personal tutoring, or pay to join a class, and that pretty much comes up September. All right, we are still going to be having live, still going to be having these free sessions, but do you understand that what makes the money, what makes the mail run? All right, so glucose is converted to starch for future use. Yeah, but Amai, we want to know, why is it converted? Why you couldn't just store glucose? Why you have to convert it? Right? That is what was answered before. All right, so you need to know that. That is what the kids did not know today, and it bold a couple of them. All right, great. So just let me move on. There's a lot of things that it is converted to, and I really don't want to get into everything. Yes, converted to glycogen. All right, great. All right, so great. I'm seeing a lot of things there. All right, so we continue with photosynthesis. So you are supposed to know the fate of photosynthesis. You need to know where it occurs. You need to be able to identify the equation and the part of the equation. You need to be able to define photosynthesis. Then we come down and say that we are supposed to be able to investigate the effect of light and chlorophyll on the production of starch. So that is a lab. And most HSB students would not have done that lab. It is a popular lab on your multiple choice paper, and I'm just going to tell you about the lab. We can't do the lab, so I'm just going to be telling you about the lab now. All right. There are some leaves like hibiscus leaf that have two colors, white and green. They are called variegated leaf. We carry out a process on them, an experiment on them to determine if the white part has starch. What do? We immerse the leaf in boiling water. Then we remove the leaf from the boiling water. We add some ethanol or alcohol in a boiling tube. We add the leaf to the alcohol. Then we place the alcohol in a hot water bath. That means the fire is turned off. You cannot put the alcohol over open flame, because if you place that alcohol over open flame, you are going to have fire. Why? Alcohol boils at 78 degrees Celsius, while water boils at 100 degrees Celsius. So what is going to happen? While you have that alcohol in that hot water, the alcohol will start boiling. Why do we put the leaf in the alcohol? We are using the alcohol to remove the chlorophyll. Now, as soon as that has been done and the alcohol would have changed color to green, we are going to remove the leaf from the alcohol, and it's going to be crispy like banana chips. Because it is dry, the water has also been extracted. We are going to place that leaf in water now, and this is the part that you need to know, really need to know. We are going to remove it from that water, place it on a wild tile, and we are going to add iodine to the leaf. The iodine is the reagent that is testing for starch. What you're going to realize is that the green part of a leaf turns blue-black. It's always going to be blue-black. All right? So it's always going to be blue-black. And the white part of the leaf will take the color of the iodine, which is red-brown. All right? The fact that the white part has taken the red-brown color, it indicates that there is no starch in the white part of the leaf. However, wherever there is blue-black, it suggests that starch is in that part of the leaf. And it's the same way you test a leaf for starch. That was on the exam today as well, how to test for starch. So I am hoping that those persons who hit CXC will remember that it's like the third lab when you are doing CXC, your SBA. All right, on the production of starch. All right, great. So that is all that you need to know about photosynthesis. It did not ask me to tell you about the light and dark phase of photosynthesis, so I am not going to be telling you about it. All right, so it wants me to explain, however, the way in which other living organisms depend on plants directly or indirectly for food. Now, this was on the exam paper today. So the thing about it, this pretty much pulls us nicely into the food chain and talking about all that. I wish I had a nice setup I could show you all of that. If I was pretty much teaching where you could see, it wouldn't seem so stressful, because you have to be flipping through the book. It's a little difficult. So just let me say this to you. I'm going to slow down a little bit. Plants. We should be telling plants thanks. Every time you walk on the road and you see a plant, you should say, thank you, plant. I'm going to try it tomorrow. With every plant that I pass, I'm going to say, thank you, plant, thank you, plant, thank you, plant. And why I'm going to say, thank you, plant, thank you, plant, thank you, plant? Because the plant is able to do what I can't do. The plant is able to capture that light energy from the sun and use it to make food. As such, the plants have been given a wonderful name. They are called producers. Now, all producers are going to be autotrophic organisms. What autotrophic means? Benedict solution is something different. Please do not confuse students. All right, Benedict solution is something different. And remember, when we get there, we will deal with it. I really don't want persons to be adding stuff to really confuse us. Benedict solution is something different. Iodine, that's what we're working with. All right, great. So all producers are autotrophic organisms. What it means is that they make food for themselves. Like an automatic car, it changes the gear itself. Once you put it into drive and you start driving, it will change the gear for itself. Great. So I don't know, I'm not going there. Just remember that all producers are autotrophic organisms. I don't want to spill out too much bio on you. All right, great. So we are now going to be looking at the relationship with producers and other organisms. Now, the producer, if we were supposed to draw a food chain, and you could just draw this in your book. We have a food chain, A, B, C, D. That's a food chain with the arrows between. The energy that the plant gets, or the plant will receive the most energy, and because it's getting that energy from the sun. As the energy moves from the plant, from left, through the consumer up to quaternary or tertiary consumer, what is happening is that the amount of energy that is passed on is reducing. And it is reducing by what we call a 10% law. It is reducing because some of the energy, I want to use lost in quote, is lost to respiration, excretion, and egestion. You can speak to that. You eat and then you sweat and you go to the toilet and whatever else, and the heat energy coming off. So you're losing some of the energy that you pretty much got in the chemical energy when you ate. All right? So what happens is that as you move from left to right, the amount of energy available to sustain life will dramatically reduce. And that is the reason why at the top of the food web, we have few organisms. As we move from left to right, the food chain is pretty much restricted to quaternary consumer,